Thank you, Dean. It's a great honour to be here today, and it's a great honour to be presenting the 2014 Garnett Passe Lecture. Before I start, I just want to make sure I make a conflict of interest statement. I've got no financial interest in any company that I mentioned today in this presentation, but my institute does receive research funding from Cochlear Limited for contract-based research. So before I start talking about the Bionics and particular cochlear implants, a brief overview, in addition to what the overview Dean supplied, about Garnett Passe. He graduated in dentistry from Melbourne University and then came to the UK and specialised in otolaryngology. He served in the Second World War in the Royal Navy, and he became a very experienced and outstanding otolaryngologist in London. And indeed, he was a frequent speaker in this society. He died at a very young age, 42, in 1952. So as Dean pointed out, there are two other important players in the foundation. And the real force behind the foundation is this woman, Barbara Slater. She was, in fact, born in South Africa. She married Garnett Passe in the UK in 1939. After Garnett Passe died, she subsequently married the stockbroker Rodney Williams, and they lived in the east coast of the US. She was passionate about establishing a foundation in memory of her two husbands, and she went through an extraordinary series of processes to make sure that realisation actually occurred. And so the foundation became active on her death in 1991. And it's one of the largest bequests ever made to otolaryngology in the world. The Institute, I am the director of the Bionics Institute in Melbourne, but formerly it was called the Bionic Ear Institute, and Graham Clark was the founding director of the Bionics Institute, established in 1984, 30 years ago. Until 2005, he was the founding director, and I took over in 2005. In 2011, we renamed the Institute the Bionics Institute to reflect the broader aspects of the work we're doing within our organisation. Graham's still very much involved with our institute, and of course he's the pioneer that took the cochlear implant in Melbourne from a university cottage industry device to a commercial device that's made a huge impact around the world. The Bionics Institute, we renamed it in 2011, has three major themes of research. Bionic hearing, we still very much work in the area of cochlear implants and technologies that will improve the hearing of the patients that suffer from a severe to profound sensorineural hearing loss. But we also work in areas of bionic vision, retinal prostheses, and central nervous system prostheses we call neurobionics, for the detection, for example, of epileptic seizure using technologies that are similar to cochlear implants. Today, of course, I'm going to mainly concentrate on bionic hearing, but at the end of the talk, we'll briefly mention our work in these other two areas of research. This is the overview of my talk. I'm going to provide an introduction to medical bionics, describe sensorineural hearing loss, and then a brief overview of cochlear implants and some of the future developments where we can see improvements coming through this technology. Talk briefly about the plastic brain, because it plays an incredibly important role in all sensory prostheses. Vestibular prostheses, they're on their way. They're not in the clinic quite yet, but they're on their way, and then sum up with conclusions. So, Bionics is essentially the combination of clinical research, engineering research, and scientific research, working at the interface. It must be multidisciplinary. And in fact, Bionics is made up of the words biology and electronics. And there's many devices that we know and we take almost for granted, but are making huge impacts around the world each day, going from heart pacemakers, spinal cord stimulators for relief of back pain, cochlear implants, deep brain stimulation for the treatment of motor disorders in Parkinson's disease. Just to give you an idea of scale, 600,000 heart pacemakers are implanted each year around the world. These are huge industries making huge impacts in human health. One of the really exciting things about working in the area of bionics is there's a lot of new technologies coming through the pipeline, some of which are still on the bench, some are in preclinical studies, and some are in clinical trials. And these are just a few. Functional electrical stimulation to allow paraplegic patients to stand for short periods of time. Deep brain stimulation for severe psychiatric disorders and chronic pain. Brain-computer interfaces to allow patients that have high-level spinal cord injury to control things as simple as turning lights off and on or performing email, or as complex as controlling robotic limbs. Vestibular prostheses, as we've already mentioned, retinal prostheses, and cortical prostheses for detection of epilepsy. They're just a small number of these bioengineered devices that will be coming through to clinical and widespread clinical and commercial application. So what do these devices look like? And here's two examples: a cochlear implant, which many of you in the room know very well, and a spinal cord stimulator that will alleviate pain in patients with severe back pain. So in the spinal cord stimulator, there's a number of electrodes that are inserted parallel to the spine, and an electrode pair is stimulated to provide some form of anaesthesia. The cochlear implant is deliberately much smaller than the spinal cord stimulator, and the reasons why we'll explain in a minute. But let's make it bigger, and let's have a look at some of the common features of these bionic devices. First of all, they all contain electrodes, typically platinum electrodes. So it's very difficult to see, but there's 22 very tiny platinum electrodes on that helical electrode array that is inserted into the inner ear. The electrodes, the platinum electrodes in this device are a lot larger, still performing the same function, safely delivering electrical charge to neural tissue. These devices all have lead wire systems. So if there's 22 electrodes on the electrode array, there's 22 very fine wires that are connecting those electrodes to the stimulator that's implanted in this device here. The stimulator electronics are by necessity encased in a hermetic case, and this is typically achieved by a titanium can. So all the electronics are contained within a titanium can that's laser welded to ensure that no fluid can get in and damage the electronics. Both of these devices have these titanium hermetically sealed cans that contain the electronic components that drive these devices. And finally, power. These are electrically active devices. They need to be powered. And this is the reason why there's differences in size. These type of devices, deep brain stimulation devices and devices for pain alleviation, have batteries implanted inside them. So they've got rechargeable batteries implanted in the device. These batteries will, over 4 to 8 years, eventually expire, and the device will need to be removed and another stimulator be replaced. The cochlear implant, because it is a sensory prosthesis that needs to monitor, in the case of a cochlear implant, receiving signals about the patient's auditory environment all the time, doesn't have a battery. It receives both power and data across a radio frequency link with the outside world. So this coil here receives not only the data describing which electrodes need to be stimulated, but it actually also receives the power to power the device. Hence, these devices can be much smaller, and that's been incredibly important for cochlear implants. It means that these devices can be implanted in children as young as 6 months of age. Implanting these devices in a child that young, in a profoundly deaf child that young, provides auditory input at a stage when the child is geared to learn language. You couldn't get a better combination. So these devices started to appear in the late 50s and early 60s. again based on the technology that others had developed. These devices are electrically active, so they need to have some form of amplification. We couldn't amplify an implantable device with a valve that were the only form of amplification before the 1960s. You can't implant valves, they require significant power and they generate a considerable amount of heat. This is in fact a photograph of the first patient with a heart pacemaker. It was on a trolley. There's a heart pacemaker. It's outside with a lead wire going into the heart. And a very stern nurse to make sure that the electrical connection to the power point wasn't pulled out. So it was a low quality of life, but it was a lifesaver for this particular individual. It was based on valve technology. But the field moved on very, very quickly. As soon as transistors became readily available, the biomedical engineers and the clinicians saw the opportunity of using devices that are low power, do not generate much heat, and can be implanted in the human body. So in fact, this is one of the first implantable heart pacemakers, circa 1965, an English heart pacemaker, with discrete components, transistors and batteries, and in this case, hermetically sealed with epoxy resin. Epoxy resin wasn't bad. It would last for about 4 or 5 years before the fluid would get in and damage the electronics. But it allowed the patient to move around in a normal, unobstructed way. The real breakthrough came in the 1970s and subsequently when transistors merged into integrated circuits. And not only 4 or 5 transistors could be put in a device, but hundreds and hundreds of thousands of transistors could be put on one chip, developing incredibly powerful devices that can not only electrically stimulate, but can also record from the biological environment and send that signal back out. And this is the heart, integrated circuits of the heart of all modern bionic devices. And this is how they're assembled. There's the integrated circuit. There's some other components on the printed circuit board. And that printed circuit board is placed between 2 halves of a titanium can, and those titanium cans are welded closed, creating that hermetic seal. And then the whole device is encapsulated in medical grade silicon. So that's an overview of how medical bionic devices are assembled. Let's look specifically now at cochlear implants and the issue around sensorineural hearing loss. So everybody in this room knows very well that sensorineural hearing loss is the most common form of hearing loss and typically involves the permanent damage to the hair cells of the cochlea. Unfortunately, as mammals, we do not have the ability to regenerate hair cells. This is the classic wiring diagram of a mammalian cochlea. So here's a scala tympani down here. These cells are the spiral ganglion cell bodies, and they have peripheral processes that project out to innervate the 1 row of inner hair cells and the 3 rows of outer hair cells in the organ of Corti. So the type 1 ganglion cells project through and innervate their closest inner hair cell, and there's about 1,800 inner hair cells in a normal cochlea. And the type 2 ganglion cells innervate several outer hair cells. They push through a bony structure called the osseurospiral lamina through these tiny holes, the habenula perforata. So this is a habenula perforata from a rat cochlea, illustrating the exquisite anatomy of the mammalian cochlea. If we take the bony structure off and look down at where the ganglion cell bodies, the spiral ganglion cells would normally reside, they reside in a structure called Rosenthal's canal. So normally this would be packed full of the cell bodies of the auditory nerve. And this is the scala tympani. This is where we'd traditionally place our electrode. And this is the wall, this thin bony wall between Rosenthal's canal and the scala tympani. This wall is very thin, it's 10 microns. It's a very thin bony shelf, but separating Rosenthal's canal and the scala tympani. If we now have a look from the scala tympani, from where an electrode array would be located, towards the spiral ganglion cell neurons, we see that it's actually a very perforated structure. And the reason I'm showing you this, it's thin and it's perforated, is that if we provide drugs to the scala tympani, these drugs have access through these so-called caniculae perforentes into the spiral ganglion cells. So we can help prevent the degeneration of these ganglion cells by providing drugs not directly to the cells, but to the fluid within the scala tympani. The organ of Corti, the hair cells of the organ of Corti, are not only releasing neurotransmitter to excite spiral ganglion neurons so we can perceive hearing, but they're also releasing small amounts of endogenous neurotrophins. Brain-derived neurotrophic factor is one of two of these neurotrophins that are being released by the inner hair cell in particular and the support cells of the organ of Corti. This neurotrophin release is vital for the exquisite wiring diagram that we've just discussed in terms of the organ of Corti. But in addition, that is during development. But in addition, the neurotrophins that are being released in a mature cochlea help provide and maintain the normal spiral ganglion cell population. So in a condition, a sensorineural hearing loss, where we've damaged the inner and outer hair cells and the support cells of the organ of Corti, as in this example here, we see ongoing loss of our spiral ganglion neurons. And the reason this interests me is the spiral ganglion neurons are our target neurons for a cochlear implant. So we're going to have a look at a little bit more detail. This is the packing density of the peripheral processes just before they innervate the organ of Corti, the inner and outer hair cells, in a normal guinea pig cochlea. The packing density is high and the diameter of the fibres, the type 1 fibres projecting to the inner hair cell, is very uniform because that conduction velocity needs to, of the action potentials need to be uniform for us to understand and appreciate audition. After 5 weeks of deafening, we see significant dieback of the peripheral processes from the organ of Corti. And in fact, here's a low power and high power image of the peripheral processes from normal through to 12 weeks of deafening. And you can see the reduction, the retraction of those peripheral processes from the organ of Corti back to the spiral ganglion cell body. This is also seen in human cochlea. So this is an image looking down on the top of a human cochlea taken by Harold Schutnick, and he stained this cochlea with osmium tetroxide so you can see the exquisite branching of the peripheral processes as they leave the organ of the spiral ganglion cell bodies and project through to the organ of Corti. And you can see the hair cells. The thick line is the outer hair cells, the thin line is the inner hair cells. You can see that extensive projection. But in this region of the cochlea, where this patient had lost hair cells, there's extensive loss of the nerve fibres. So what we see in an accelerated form in experimental animals is also true in the human cochlea when hair cells are lost. The spiral ganglion cell bodies, the cell bodies themselves, also degenerate over time. They degenerate relatively rapidly in experimental animals, but much, much slower in humans. So again, this is a low power of a normal packing density of the spiral ganglion neurons and the reduction over a 12-week period following sensorineural hearing loss. So as we remove the source of these neurotrophins at the organ of Corti, we're getting some dieback of the neurons that we're trying to stimulate with a cochlear implant. Again, this is seen in human material. And this is work by Joe Nadel, where he's published cadaver material where he's counted the entire spiral ganglion cell population for normal patients. A normal human has about 30,000 spiral ganglion cells and various etiologies. And we can see that etiologies where there's either been a long-term duration of deafness or an invasion via infection, such as bacterial or viral meningitis, exhibit the most extensive forms of spiral ganglion cell loss. So in many cases, patients receiving cochlear implants might only have 10 to 20% of their normal spiral ganglion cell population. So just a summary, what we see with loss of what occurs after the loss of the organ of Corti is that we see an extensive loss of the peripheral processes that would normally innervate the organ of Corti. We get a shrinkage and a gradual loss of the spiral ganglion cells, our target neurons with the cochlear implant. I haven't had time to talk about this, but we get shrinkage of cells within the central auditory pathway, but importantly, we don't see any evidence of degeneration of cells within the central auditory pathway. And also importantly, these partially degenerated spiral ganglion cells still remain capable of initiating and propagating action potentials through electrical stimulation. And finally, let's remember that it wasn't so long ago that a profoundly deaf patient would get a letter like this. So this letter was written by the otologist to a profoundly deaf patient saying, you will have to make the most of a pretty bad lot. No treatment was available in the 60s up to mid-70s for a profound sensorineural hearing loss. So that opened up the pathway for the development of cochlear implants. And the pathway within the mid-70s was absolutely ripe. We had a very, very good understanding from fundamental studies of the anatomy and physiology of the cochlear and the central auditory pathway. There'd been excellent proof of principle clinical trials conducted at a number of sites around the world. As we said before, integrated circuits were now becoming available, so the technology was ripe. There was absolutely a clinical need, and very importantly, there was no competing therapy. So come the mid-70s, there was about 11 research laboratories around the world developing these type of technologies. I've made the star here in Minneapolis, St Paul, large because that's where 3M had already, by 1978, gained FDA approval for their single channel device. So there's a large number of groups around the world in the mid-70s developing these technologies, including groups led by Professor Fortune in London. This is a group that Graham Clark set up in the mid-70s, well, in the early 70s. This is a strange-looking mixed match of people that represented the Department of Otolaryngology at the University of Melbourne. Most people were convinced that this mismatch of individuals could create nothing. Here's Graham Clark, and there was only one other otologist. But importantly, Graham understood the importance and principle of multidisciplinary research. So there's physicists, engineers, there's even me over here as a biologist with a little bit more hair than I have now. There's the current head of the science and technology at Cochlear, Jim Patrick, for those people that know him. So he developed this multidisciplinary team, but it was very, very difficult because there was experts all over the world saying that electrical stimulation of the cochlea just wouldn't work. So when we wrote peer-reviewed grants, we had this type of information thrown back at us. So Graham was a very enterprising fellow, and he set up with a local television station a telethon. And I vividly remember going around with a can, shaking for money in the can for dollar donations to drive this research. And this is a cartoon from a local newspaper depicting Graham Clark shaking the can to drive cochlear implant research in Australia. So without the issues of funding, we still got on and developed a device in Melbourne, a cottage industry device completely manufactured in the university using very good electronics encased in epoxy resin as a hermetic seal. And in 1978, we implanted our first of 3 patients. We called this device an aid to lip reading. we absolutely never envisaged that patients could use their mobile phones to conduct a usable conversation with these devices. All three devices in these three patients ultimately failed. And they mainly failed because of a lack of hermetic sealing, fluid getting in, damaging the electronics. But we learned a lot from those failures. The Australian government became our venture capitalists. They invested $2 million over a few years because they saw the feasibility of this technology in terms of being able to aid communication. They married our university-based research with a heart pacemaker company in Sydney called Telectronics, and they had, for the first time, developed this titanium hermetic seal, and this is their heart pacemaker in 1977 with a titanium hermetic seal. So all our failures could be overtaken by their technology. So it worked very, very well. And for over 2 years, this is the first of cochlear implants that were developed, and we got additional biological studies approved, and by 1985, FDA approval. And this is a modern cochlear implant, one of a number of different companies. In total, the three major companies have implanted over 330,000 patients around the world with these devices. And the devices, as most of you know, have an electrode array implanted in the cochlea. When we stimulate the apical electrode, patients perceive a low-pitch sensation, and when we stimulate more basal electrodes, patients perceive high-pitched sensations. So we're using that classic tonotopic organisation of the cochlea. I should also acknowledge that there are clinical devices, particularly devices that are placed on the cochlear nucleus, to also provide auditory input for patients. And again, this cochlear nucleus device is FDA approved. And there are other research programs dealing with trying to develop devices where we directly stimulate the auditory nerve, the inferior colliculus, or the auditory cortex. Those devices are experimental in nature. So future developments. These devices are not perfect. They perform very well in quiet, but they're certainly not perfect. And some of the areas of future development include very much improved performance in both noise and music appreciation. And we're looking at current focusing technologies and improving the electrode neural interface, because we had this issue of the neurons dying back away from the cochlea. We want to try and reverse that. We want to improve the performance in patients where they're implanted with both a cochlear implant and a hearing aid. Those patients perform extremely well. However, a number of them lose their hair cells over time, and their performance is reduced. So one of the major areas of research is to investigate ways where hair cell survival can be maintained. And integrating the hearing aid and the cochlear implant processing. At the moment, they're two completely separate devices. Bilateral cochlear implants, there's a lot of work being done in bilateral cochlear implants, and again, the integration of their processing strategies needs to be developed, and techniques to reduce the inflammation response around the electrode in the cochlea following cochlear implantation. So just to give you a feeling for music appreciation with these devices, this is a simulation. We don't actually know if patients would hear something like this. So I'm not sure about you, but I know I wouldn't pay good money to go to a concert to listen to that rubbish. There's a lot to be done with cochlear implants. Here's the... So music appreciation and improving the performance in a noisy environment are key areas. So that's what I'm going to talk about today in future studies. Current focusing technology and improving the electrode neural interface. So cochlear implants typically use what we call monopolar stimulation, where there's one large extra cochlear electrode and we stimulate that against each of the implanted electrodes. It's a very efficient way of stimulating, but the problem is it recruits a relatively broad number of auditory neurons. It's not incredibly focused. And that's the reason why at the moment there's no point in adding any more than up to 20 electrodes in the cochlea because of this spread of excitation. You can improve it a little bit by using bipolar stimulation, where we stimulate against adjacent electrodes within the cochlea, and that causes a slight improvement in focusing the extent of neural excitation. This is a diagram of the inferior colliculus. This is a structure within the auditory midbrain, and like all structures within the auditory pathway, it is tonotopically organised. So it's organised from low frequencies in the base to high frequencies in the low frequencies to high frequencies. And as we put a multiple electrode array in, we can record from different sites within this low to high frequencies region and determine very extensive fine tuning curves for auditory stimulation. So this is a 12 kilohertz tuning curve, and it excites a very localized region within the inferior colliculus because of this highly frequency organized structure. We use this to determine the extent of excitation with electrical stimulation. So we put electrodes in rather than acoustic stimulation, and we can do the same experiment where we map the inferior colliculus and look at the point of lowest threshold. And for monopolar stimulation, we're seeing very little tuning compared with the type of tuning one receives naturally through auditory information. And that excitation spreads right throughout the cochlea. So this is one of the problems we face. When we're using two new different types of stimulation strategies, tripolar stimulation and focused monopolar, we can actually really finely tune the cochlear implant excitation field. And we believe that if these devices can get into clinical application, they'll be able to provide patients with far more information about timing and pitch that will be necessary to understand human speech in a noisy environment. So what is tripolar stimulation? Tripolar stimulation is stimulating between one central electrode and two flanking electrodes, as shown here, and it produces more localized field of excitation. Focused monopolar is a very complex form of stimulation. In fact, this technology was taken from the field of radar physics, where we actually stimulate several sites simultaneously to force a very, very localized field of excitation. So these technologies are on their way to trying to be developed for clinical application. And they certainly do produce far more localized fields of excitation with electrical stimulation, as indicated here. But our big problem at the moment is they're inefficient. They require far more stimulus than a monopolar stimulation. So one of our major areas of research is to investigate how these technologies can be improved, but be also very, very efficient. And I must point out that we're also undertaking a clinical trial in this work at the moment, with Garnett Passe leading that. And all this work has been funded by the Garnett Passe. The other area that I hinted about was the issue of improving the electrode neural interface. So this is the normal cochlea, or normal organ of Corti, spiral ganglion cells and peripheral processes. In the deafened cochlea, when we've removed or damaged the organ of Corti, we get this dieback of the neurons from our electrode array. And we're looking at techniques to overcome that. We know that when we place an electrode into the inner ear and stimulate, that depolarization will not actually reverse this degeneration process. But we also know that when we provide exogenous form of neurotrophin in combination with the electrode, we get massive evidence of rescue. So rather than the neurotrophins being supplied by the hair cells that no longer exist, we're supplying them artificially. Here's an example. So this is the cell bodies of hearing nerves in an animal deafened for 4 weeks. And the next slide is the opposite cochlea of this animal. Normally, they'd show extremely symmetrical loss of auditory nerves. But in this cochlea, we treated with neurotrophins. And there's normal nerve survival. Absolutely normal nerve survival. And in addition, there was a reduction in electrical threshold. So there's functional benefits as well as anatomical benefits. So over the past few years, we've been investigating ways of safely delivering these drugs into the inner ear. And we thought about pumps, but we don't like pumps. We don't like cannulas going into the cochlea because of issues around infection. So we've turned to nanotechnologists, creating small particles that will release these drugs over a period of time. We've looked at cell-based therapies and gene therapies. And I'll take you through a couple of examples at the moment. So working with nanotechnologists, they have assembled these mesoporous silica particles that, when assembled, consist of a diameter of about 0.5 millimetre and can be loaded with not only BDNF, but from an etology point of view, you can put any number of different therapeutic drugs into these capsules. We've looked at the release of these drugs, and we can get good release properties over more than 60-day period of time. And that's the type of time we believe is necessary to release these drugs to get good efficacy. And we've developed the surgical techniques to implant them. They're 0.5 millimetre, so they readily can be implanted via a cochleostomy into the scala tympani, and they evoke a minimum tissue response. You can only see a very fine fibrous tissue reaction there. So when we look at these particles, you can see excellent, after 1 month of implantation, excellent nerve survival in the BDNF particles compared with the control particles, deafened for the same amount of time, and there's the statistics. So not only could this technique be used for delivering neurotrophins, but it could also be used to deliver other therapeutic drugs into the inner ear. The other interesting area is what we call cell-based therapies, where we pack cells that are either artificially releasing neurotrophins or are genetically modified to release neurotrophins into an alginate capsule. So the neurotrophins can be released, but the cells within the alginate capsule are not evoking an adverse tissue response within the biological environment. And here's some examples of 0.5 millimetre diameter alginate capsules with cells excreting neurotrophins. We've developed, again, a surgical technique to implant them. We can implant up to about 10 in the guinea pig cochlea. And this is an example of one of these catheters going into the cochleostomy of a guinea pig. And this is the concept. So we have an electrode array, we have our capsules releasing neurotrophins, all in the scala tympani. Again, we've done animal studies, and in this case, the animal studies were performed in cats. They were deafened for 8 months, so these are the controls. 8 months of deafening in a cat results in very few ganglion cells. There's only about 6 ganglion cells left in this control cochlea. Whereas there's a lot more ganglion cells in the side that was treated with the neurotrophin, the cell-based therapy. And statistics to prove that we could get significant rescue effects with neurotrophins alone and neurotrophins with cochlear implantation. So these are potentially clinically viable technologies that can be used to deliver a number of different therapeutic drugs into the inner ear. Our interest is delivering neurotrophins. So what we obviously want to do is reverse this dieback by providing neurotrophins so that the neurites will grow close to the electrode or even onto the electrode and provide a far more efficacious device where stimulus levels will be a lot lower. So very briefly, the plastic brain. We know that the auditory cortex contains that same tonotopic map as the cochlea, low frequencies to high frequencies, just like every other structure within the central auditory pathway. What we don't know is the status of the auditory cortex in a child or an animal that's been deaf since birth. Does auditory experience need to drive this process? So we've performed a number of animal studies where we've had cats actually wearing real cochlear implants. They've been deafened at birth and they've received their only auditory input through a cochlear implant, and they've been stimulated for approximately 6 months. So here's an example of that's our cat with the device off, just to prove that they can hear. So they can certainly attend to their auditory environment through use of a cochlear implant. And at the end of the experiment, we examined the brain structure in the auditory cortex using these multi-channel electrode arrays. In fact, these are the same electrode arrays that groups are using in humans as brain-computer interface electrodes. So we implant them in the auditory cortex, and we can monitor the neural profile of the auditory cortex using this type of technology. And this is a summary of what we've found. So cochlea has high frequencies in the base, low frequencies in the apex, and in the normal auditory cortex, we get that exquisite cochleotopic organisation. In a long-term deafened animal, something that's had no auditory experience, the auditory cortex is completely scrambled. There's no evidence of this exquisite cochleotopic organisation. And what we've found with animals that have been chronically stimulated with these devices, either through early cochlear implantation or receiving stimulation at a later age, is that we can return this cochleotopic organisation to near normal structures. So the importance of auditory input via acoustic or even electrical drive from the cochlea plays an important role in returning and maturing the central auditory pathway. And this also is seen in every cochlear implant clinic around the world, as almost all cochlear implant patients show ongoing improvement of their hearing experience with device use. The plastic brain is a major factor in the success of sensory prostheses like cochlear implants. As a biomedical engineer, I know that we're providing an incredibly crude stimulus to the auditory nerve. We're trying to reproduce hearing that would normally be coded onto 1,800 inner hair cells with less than 20 electrodes. It's an incredibly crude stimulus. The plastic brain can take that stimulus up and attend to it, provided we, with this device, can provide behaviourally relevant information. So vestibular prostheses, I'm far from an expert on vestibular prostheses. You've seen this map before. This is the activity that I know of, at least, with vestibular prostheses, mainly associated in Europe and the US. And there's two types of devices that are in development. The stars indicate, to my knowledge, devices that have already gone into clinical trial. So there's devices simply based on cochlear implant technology, where the electrodes are inserted rather than the cochlea into the semicircular canals, that these devices do not sense head movement. They are there for a Meniere's attack, and then the patient can activate the device based on a Meniere's attack. Then there are devices that are more sophisticated that sense head movement using a variety of different gyroscopes. And these devices are designed to detect orientation and acceleration. So the first of these devices was recently implanted in the Netherlands last year. There's a large project undertaken by the European group, and there's a group in Johns Hopkins developing that technology. So we need to watch this space. It's on its way. And finally, conclusions. So what's the Garnett Passe meant to me? I received my Garnett Passe Senior Principal Research Fellowship in 1996, and it provided me with the ability to do my own research, set up my own research lab and my own research program. It was a groundbreaking experience for me. I was also very privileged to have a number of UK and Irish otolaryngology collaborators, and they're named there, and I'm sure some of you know some of these guys. And also the privilege of supervising 24 research students, including PhDs. So we were able to undertake the research I just talked about, but we're also able to undertake other research, including research more recently in the bionic eye. And this is a brief video of our first bionic eye patient. My lab designed the electrode for the device, did all the preclinical safety studies, and worked closely with the retinal surgeons to develop the surgical procedure. And this woman is, and two other patients are now using their devices in a restricted clinical trial.
It was really funny when it was switched on. I was, waiting. I had these goggles on, and I didn't know what to expect. It was just, and I don't know if anyone did know what I was going to see. And then all of a sudden I went, yep, I could see a little flash, and it was like a little, I suppose, a splinter, and there were different shapes and dark black and lines of dark black and white lines together. Then that turned into splotches of black with white around them and cloud-like images. It was just, I can remember when the first sort of bigger image came there, I just went, wow, because I just didn't expect it at all. It just, but it was amazing.
So she was featured on the Australian National News yesterday. She's now using this device with a video camera in a low vision laboratory, and she's able to identify obstacles and move around obstacles. There's still a lot to be done, but we believe we are providing that behaviourally relevant information, and hopefully these devices one day in the future will be successful. We're also working in the area of what we call neurobionics, and we're taking part in a clinical study where we're using DBS to treat obsessive compulsive disorder, refractory to any drug-based therapy, and that work is going very well. So in conclusion, I'd like to acknowledge my collaborators, both within the Institute and the research collaborators, and our clinical collaborators, our funding, and in particular the Garnett Passe and Rodney Williams Memorial Foundation. Thank you very much.